Welcome to AMiNDR, a podcast where we summarize the latest publications on neurodegenerative disease research so that you can stay up to date with the newest findings. Every month, our team of scientists will sort and organize the titles into themes and present shortened versions of the abstracts. We'll make sure to mention the title, the journal, the first author, and the last author for each publication. Whether you're in the lab, on the bus, or cooking your meal, we hope you find this podcast helpful.
Hello, this is Naila, and I'll be your host for today's episode. So today we're covering papers that were published in June 2020 on non-pharmacological approaches to treating AD. So I've separated this into 4 sub-themes. The first will be 5 papers in the diet and microbiome sub-theme. Then we've got 8 papers on physical exercise, 2 papers on other behavioral interventions, and 6 more papers on what I've decided to call physiological interventions. So these are medical interventions that are not pharmacological in nature. So let's get started with the diet and microbiome. So the very first paper I'll discuss in this category is on intermittent fasting. This is interesting to me because at the beginning of the pandemic, when we were all in lockdown, I actually tried a 60-hour fast. It was, I will admit, mostly out of boredom and just wanting to challenge myself, but to feel more scientific about it, I read some review articles beforehand, and I was really interested in how intermittent fasting might be protective against neurodegenerative disorders. So I'm not sure I'll be trying that again, but at least this next paper makes my suffering during that time a little more justified. The title is Intermittent Fasting Promotes Adult Hippocampal Neuronal Differentiation by Activating GSK3 Beta in 3X Transgenic AD Mice. This was published in the Journal of Neurochemistry by first author Lee and last author Wei. Moderate dietary restriction could be protective by promoting neurogenesis and increasing the expression of neurotrophic factors. Glycogen synthase kinase 3 beta, so that's GSK3 beta, signaling is crucial in neurodevelopment, and it's possible it could play a role in dietary restriction. This is what the authors wanted to find out. They had 4 groups of 6-month-old mice in order to test their research question. These were separated by genotype, so wild-type or 3X transgenic AD mice, and also by diet. So either intermittent fasting, fed on alternate days, or ad libitum, so free-for-all essentially. They ran this diet for 3 months and found that it promoted the differentiation and maturation of neurons in the dentate gyrus of the hippocampus and improved the cognitive dysfunction normally seen in transgenic AD mice. Intermittent fasting inhibited the insulin and protein kinase A or PKA signaling pathways, while activating the AMP-activated protein kinase and brain-derived neurotrophic factor pathways. So that's AMP protein kinase and BDNF pathways. But crucially, when the authors infused GSK3 beta siRNA, so this is in order to downregulate GSK3 beta activity, when they infused this in the dentate gyrus during the diet period, this reversed the positive effects seen in the fasting mice. So overall, these findings suggest that intermittent fasting could improve learning and memory, and this is by activating GSK3 beta, which in turn promotes neuronal differentiation and maturation in the hippocampus. Alright, next up we have a paper on something I've never heard about, which is hairy garlic. Its scientific name is much more complicated. I'll give it a go. It's Allium subhirsutum. And this is a bulbous plant that's widespread in the Mediterranean area and is locally used as food and spice. So the title of this paper is Hairy Garlic, or Allium subhirsutum, from Sicily, Italy. LCDAD-MSN analysis of secondary metabolites and in vitro biological properties. This was published in Molecules by first author Sutt and last author Dall'Acqua. And the study characterized the chemical profile of the ethanolic extracts from this garlic's bulbs and aerial parts, and evaluated their antioxidant properties. They also examined whether the plant has any acetylcholinesterase inhibitory properties, which could be particularly relevant for AD, and indeed they found inhibitory activity in both the bulbs and the aerial plant extract. Both extracts also exhibited moderate antioxidant properties in vitro assays, and the authors report that hairy garlic is a good source of sulfur compounds, flavonoids, and phenylpropanoid derivatives, making it a valid alternative to the common garlic. So maybe that is the secret to the healthy Mediterranean diet. Okay, moving on to a different geographic location and a different food. This next paper is on high-altitude Tibetan fermented milk, and it touches on what has become a hot topic in neurodegenerative disease research, namely gut microbiota, and how we can have more of the good kind in order to promote brain health. So this paper is entitled High-Altitude Tibetan Fermented Milk Ameliorated Cognitive Dysfunction by Modified Gut Microbiota in Alzheimer's Disease Transgenic Mice. This was published by first author Liu and last author Cui, or CUI, and it was in the Journal of Food and Function. This study investigated whether Tibetan fermented milk, which is similar to yogurt, could affect memory impairment in the APP-PS1 transgenic mouse model of AD, and whether this is by changing the gut microbiota. They used Tibetan fermented milk, selecting for the samples with the highest microbial diversity as an intervention in these AD mice, in complement to their basic diet, which was an aseptic maintenance feed. After 20 weeks of mice eating fermented milk, fecal samples were collected for 16S rRNA sequencing, and the authors examined cognitive function in the mice as well as A-beta deposition, so amyloid beta deposition, in the hippocampus and cortex. They found that Tibetan fermented milk improved cognitive impairment in the APP-PS1 mice, including both spatial and object recognition and memory. And it's a little unclear in the abstract what the control group was for this, so you'll have to check the paper if you're interested. The fermented milk also increased intestinal microbial diversity and elevated the relative abundance of certain microbiota. I can't pronounce these, so check the paper for the names. And also in the paper, you'll find which ones were correlated negatively or positively with cognitive function. The authors also report that Tibetan fermented milk reduced amyloid beta deposition in the cerebral cortex and hippocampus, and they conclude that its long-term intake can have a beneficial effect on the composition of intestinal flora and on cognitive function, at the very least in these AD mice. So sticking with microbiota, this next paper focuses on the metabolites produced by intestinal microbiota, specifically on short-chain fatty acids, which have been implicated in immune function, host metabolism, and even in behavior. The title of this paper is Effects of Probiotic Supplementation on Short-Chain Fatty Acids in the APP-NL-GF mouse model of Alzheimer's disease. It was published in the Journal of Alzheimer's Disease by first author Kaur and last author Combs. So the authors wanted to know whether administering probiotics can influence levels of intestinal microbiota and their metabolites, and whether this in turn could improve pathological changes in the mouse model of AD. They compared wild-type mice to APP-NL-G-F mice, I'll just refer to these as AD mice, and treated them with either a vehicle or a probiotic, which they call VSL number 3. After 8 weeks of treatment, they analyzed the fecal microbiome, as well as a variety of short-chain fatty acids in the brains and serum, and they also looked at immunohistochemical levels of amyloid beta, GFAP, IBA1, CFOS, and Ki67. The probiotic supplementation altered the microbiota in both wild-type and AD mice and increased the serum levels of short-chain fatty acids acetate, butyrate, and lactate. Surprisingly, however, when analyzing brain samples, the authors found that the probiotic only increased lactate and acetate in the AD mice. While most of the immune markers were unchanged, there was also an increase in hippocampal CFOS staining, specifically in the AD mice that were fed the probiotic. Overall, these findings show that probiotic supplementation has differential effects on the microbiome in this AD mouse model compared to wild-type mice, and that some short-chain fatty acid changes correlated with the increased expression of a neuronal activity marker. So I'd be interested in seeing a follow-up paper in which they describe the behavioral and cognitive consequences of these microbiome changes. All right, and this last paper in the diet and microbiome section will transition nicely into our exercise section, as it examines whether exercise can improve gut dysfunction and in turn alleviate neuropsychiatric symptoms caused by amyloid beta. So the title of this paper is Prophylactic Effect of Physical Exercise on Amyloid Beta 1 to 40 Induced Depressive-like Behavior and Gut Dysfunction in Mice. This was published by first author Rosa, last author Rodriguez, and others in the Journal of Behavioral Brain Research. The authors tested whether treadmill exercise for 4 weeks, 5 days a week and 40 minutes a day, could counteract the depressive-like behavior, the altered duodenum morphology, and the abundance of Firmicutes and Bacteroidetes. I even just looked up how to pronounce that and forgot. Bacteroidetes? I don't know. Anyway, those two phyla that are all induced by amyloid beta 1 to 40. So intracerebroventricular administration of amyloid beta 140 in mice increased the immobility time in the tail suspension test, indicating behavioral despair, and it also reduced time spent sniffing in the female urine sniffing test, which suggests impaired reward-seeking behavior. This was accompanied by reduced villus width and abnormal epithelial microvilli in the duodenum. There were also other A-beta-induced changes in the duodenum, but you'll have to check the paper for details. I have, however, looked up a very useful YouTube video on how to pronounce Bacteroidetes and Firmicutes. I've been practicing, so now I can tell you that these amyloid beta injected animals also presented with reduced Firmicutes and increased Bacteroidetes abundance, which led to an increased Bacteroidetes to Firmicutes ratio. I'm still struggling. But anyways, with regards to physical exercise, the authors state that, and I quote, most of the alterations observed in amyloid beta 1-40 exposed mice were prevented by the practice of physical exercise. Well, I guess you'll have to read the paper to know more. So that's it for that section. And next up, we have physical exercise. So in this subtheme, we have 8 papers on physical exercise, and we'll start with a few in animal models before moving on to human research. The first paper in this section examined whether swimming exercise could improve memory impairment, anxiety, and depression-like behaviors in a mouse model of AD. This was published in the Journal of Physiology and Behavior by first author Bashiri, last author Salari, and the title of this paper is Swimming Exercise Improves Cognitive and Behavioral Disorders in Male NMRI Mice with Sporadic Alzheimer-like Disease. So in this study, mice were injected with streptozotocin to induce an Alzheimer's-like phenotype, and 8 days later, they were subjected to swimming exercise for 4 weeks. Numerous behavioral tests were conducted to assess cognitive function as well as anxiety and depression-related behaviors, and the authors measured several protein levels within the hippocampus to determine a possible mechanism. They found that streptozotocin impaired cognitive functions while increasing anxiety and depression-like behaviors and elevated the levels of amyloid beta 42, glutamate, malondialdehyde, TNF-alpha, and interleukin-6 levels in the hippocampus of these mice. Swimming exercise was able to reverse the neurobehavioral effects and decrease the levels of glutamate and TNF-alpha while increasing BDNF. Overall, this suggests that swimming exercise could be beneficial in reducing neurobehavioral disorders associated with Alzheimer's. This next study looked at whether antioxidant supplementation is a viable complement to exercise for improving cognitive and motor performance in an AD mouse model, namely the ApoE3 and ApoE4 transgenic mice. The title of this paper is ApoE Genotype-Dependent Response to Antioxidant and Exercise Interventions on Brain Function. The paper was published in Antioxidants, Basel Journal, and the first author is Chowdhury and the last author is Sumian. The authors examined 12-month-old male and female mice expressing human ApoE3 or ApoE4, so that's apolipoprotein, and fed them either a control diet or a diet supplemented with vitamins E and C. They further separated the mice into a sedentary group or daily exercise group. After 8 weeks on the treatments, a battery of functional tests was conducted to measure motor, cognitive, and affective behavior, while the mice remained on the treatment. Afterwards, the authors measured plasma inflammatory markers and catalase activity in various brain regions. They found that the ApoE4 mice exhibited poorer motor function and spatial learning and memory, and were not responsive to exercise or antioxidant treatments. However, the treatments did improve balance, learning, and cognitive flexibility in the ApoE3 mice. While there were no antagonistic interactions between the antioxidant supplementation and training regimen, the only further improvement was in the active avoidance task. These findings suggest that there is a window of opportunity for treatment and that genotype is an important consideration in the response to these interventions. Sticking with ApoE, I'll briefly go through a paper that announces a future randomized controlled trial on exercise. So this paper is called The Effect of Physical Exercise on Cognition Relative to ApoE Genotype, in brackets PAAD2, study protocol for a phase 2 randomized controlled trial. And this is by first author Park and last author Etnier, that's Etnier, and this was published in BMC Neurology. And as a reminder, you can always find all of the papers we mention in the bibliography, so I'm sorry for butchering a lot of names, but hopefully you can easily find these. All right. So this randomized clinical trial has 3 aims. The first aim is to test the causal relationship between physical activity and AD-associated cognitive function for persons with a history, specifically a family history of AD. The second aim is to determine whether ApoE4 carrier status plays a moderating role on cognition. And the third is to assess cerebral structure and function, as well as putative biomarkers as mediators of the effects of physical activity on cognition. The authors are recruiting cognitively normal, middle-aged, sedentary adults with a family history of AD, will determine their ApoE genotype from a saliva sample, and will randomly assign them to either a 12-month physical activity intervention or to a control group maintaining their normal lifestyle. They will conduct a variety of cognitive tests pre, mid, and post-intervention, and will also assess brain imaging and blood biomarkers pre and post. They hypothesize that 1. Physical activity will improve cognition, 2. This improvement will be moderated by ApoE4 status, and 3. That physical exercise will also cause changes in neural and blood biomarkers that contribute to cognitive changes, and that this will differ as a function of ApoE4 status. So I guess we'll have to check back in a couple years to see the results of this trial. But in the meantime, here's another future randomized trial in the planning, this time on promoting physical exercise for the prevention of AD in adults with Down syndrome. So this was published in Contemporary Clinical Trials Communications by first author Tomi and last author Donnelly, and the title of this paper is The Promotion of Physical Activity for the Prevention of Alzheimer's Disease in Adults with Down Syndrome, Rationale and Design for a 12-month randomized trial. So I actually just recently learned that nearly all individuals with Down syndrome show AD-associated pathology, and apparently this can occur as early as 30 years of age. As you've heard by now, there's increasing research suggesting that moderate to vigorous physical activity could improve cognitive function and protect against age-related brain changes, but it hasn't really been tested in adults with Down syndrome. Participation in mild to vigorous physical activity, I'm just going to call this physical activity for this paper. So participation in physical activity among adults with Down syndrome is low, and the limited research evaluating strategies for increasing exercise in these individuals has shown that most have been unsuccessful. The authors previously reported on a remotely delivered real-time physical activity intervention led by a trained health educator for adults with Down syndrome, which was highly attended, increased exercise during the group sessions, and led to improved cognitive function. Now they propose to evaluate further measures of this remotely delivered intervention in 80, meaning the number 80, non-demented adults with Down syndrome, including its sustainability and efficacy, and the impact on total physical activity, daily physical activity, that is, cardiovascular fitness, quality of life, cognitive function, and brain parameters related to AD. Next, here's a paper on a completed pilot randomized controlled trial called the RESIDENT study, which stands for Reducing Sedentary Behavior Among Mild to Moderate Cognitively Impaired Assisted Living Residents, a pilot randomized controlled trial. So this was published in the Journal of Aging and Physical Activity, and this is a one-author paper, namely Dylan, the author's name, as in last name. Okay, so the primary purpose of this pilot study was to assess whether a prompting device could reduce sitting time with light walking among older adults with mild to moderate cognitive impairment in an assisted living setting. As a secondary measure, the effectiveness of the intervention on the residents' cognitive function, physical function, and quality of life was evaluated. 20 participants with a mean age of 87 years were assigned in clusters into a single-site pilot randomized controlled trial lasting 10 weeks, in which the intervention group was prompted with a watch to interrupt sedentary behaviors and engage in 10 minutes of light physical activity, so this was mostly walking, 3 times a day, specifically after meals. Adherence to the intervention was high, with over 70% of participants completing over 80% of the prescribed physical activity bouts. There were, as the author puts it, significant effects favoring the intervention in all the measured outcomes, which included a cognitive AD assessment scale, a timed up-and-go motor task, and a short-form health survey. Next is a paper that reports back on an observational prospective cohort study on physical exercise in a medical center. This was published in Medicine Baltimore by first author Chen and last author also Chen. In fact, 4 of the authors on this paper have the last name Chen, 4 out of 6 that is. And the title of this paper is The Impact of Exercise on Patients with Dementia, a 2-year follow-up. So in this study, 80 older persons with mild to moderate Alzheimer dementia were followed up for over 2 years. The study participants were divided into exercise and no exercise groups based on their weekly exercise habit, and mini mental state examination, so that's MMSE, clinical dementia rating, which is CDR, and senior fitness tests were checked at onset. The authors defined death and unexpected hospitalization as the outcomes. They found that age, sex, education years, and MMSE showed no significant differences between the groups in all study participants. Everyone in the exercise group showed significant improvements in strength, endurance, and balance, and exercise was also associated with better senior fitness test scores. Overall, there were no differences in hospitalization and mortality compared to the non-exercise groups. However, there was some difference in the effects of exercise between the mild and moderate dementia groups. For instance, there were less unexpected hospitalizations in the exercise mild dementia group compared to the no exercise group, but you'll have to check the paper for further details. Now we'll turn to a study that looked at whether moderate intensity exercise changes cortical thickness and subcortical volumes in preclinical AD patients. This was published in Psychiatry Investigation by first author and last author Lim, and the title of this paper is Effects of Moderate Intensity Exercise on the Cortical Thickness and Subcortical Volumes of Preclinical Alzheimer's Disease Patients, a pilot study. So this study enrolled 63 preclinical AD patients with MRI and 18-fluorobutadiene PET imaging data and attained additional information on demographic characteristics, cognitive battery scores, and self-reported exercise habits. The authors found that, compared to the exercise group, the non-exercise group had reduced cortical thickness in a long list of regions that made up about half of this abstract. You can check the paper for details, but I'll also tell you that volumes of the right thalamus, caudate, putamen, pallidum, hippocampus, and amygdala were also reduced. So it appears that moderate intensity exercise affects cortical and subcortical structures in preclinical AD patients, which could have important implications for cognitive function. But it would be interesting to test this in randomly assigned groups rather than basing it on an exercise self-report. And the last study in this section examines how inter-individual differences in the response to aerobic exercise might account for the inconsistent effects on cognition, particularly in individuals with AD. The title of this paper is Inter-Individual Differences in the Response to Aerobic Exercise in Alzheimer's Disease, Findings from the FIT-AD Trial. The first author is Yu, and the last author is Mathiasson, and this was published in the Journal of Sport and Health Science. The authors examined the inter-individual differences in aerobic fitness and cognitive responses to a 6-month aerobic exercise intervention in community-dwelling older adults with mild to moderate AD dementia. They measured aerobic fitness with 3 different tests: the shuttle walk test, that's SWT, a 6-minute walk test, that's 6MWT, and maximal oxygen consumption test. And they also evaluated cognition using the ADAS-COG scale. Inter-individual differences were calculated as the differences in the standard deviation of 6-month change on the test scores between the intervention and the control groups. The authors found that there are true inter-individual differences in aerobic fitness and cognitive responses to aerobic exercise in older adults with mild to moderate AD dementia, and that these could indeed underlie the inconsistent findings of cognitive benefits in human studies. So that wraps up our physical exercise section, and I'll be back in a moment for the next section. And next up, we have 2 papers on behavioral interventions. So these are 2 papers of possible interventions for improving cognitive function. I'll be brief with the first one as it's not really related to AD per se and would actually fit better under modifiable risk factors, but I recorded that episode earlier this month. So this was published in American Journal of Geriatric Psychiatry. The title of the paper is The Association of Late Life Cognitive Activity with Healthcare and Financial Decision-Making in Community-Dwelling Non-Demented Older Adults. And this is by first author Glover and the last author is Boyle. So this study tested whether late-life cognitive activity is associated with decision-making in older adults, and whether this association varies by level of cognitive function. Using a cross-sectional design, the authors collected data on late-life cognitive activity, cognitive function, and decision-making from over 1,000 older adults without dementia living in community-based residences. Anyways, after adjusting for age, gender, and education, the results showed that frequent engagement in late-life cognitive activity can help maintain decision-making among older adults, and this is particularly true among those with lower levels of cognitive function. And the second paper is on musical and sensory motor intervention for people with AD, which is actually right up my alley. So, this was published in a French journal called Gériatrie et Psychologie Neuropsychiatrie du Vieillissement by first author Guillain and last author Sampson. And the title of this paper is Sensory Motor Synchronization and Nonverbal Behaviors in Alzheimer's Disease: The Influence of Social and Musical Context. So, increasing research suggests that musical interventions could improve the socio-emotional and cognitive functioning in adults or people with AD, particularly when participants are also engaged on a motor level. This study evaluated what factors influence the socio-emotional and motor engagement of people with AD during musical activities and measured their sensory motor synchronization abilities. Participants were asked to tap with a metronomic or a musical rhythm alongside a musician who performed the task with them. The presence of the musician was either real, so in person, or virtual. So this would be, either the live condition or the video condition. The authors found that participants spontaneously produced more rhythmic movements in response to the music than to the metronome, but the consistency and accuracy of sensory motor synchronization was actually better with the metronome, and surprisingly, it was better with the video rather than the live condition. The tempo of the auditory sequence, whether slow or fast, modulated these effects. These results confirm that musical context and social interaction play an important role in hand sensory motor synchronization, as well as in spontaneous motor and socio-emotional behaviors, and the study presents a unique multimodal approach for evaluating participant engagement in a musical task. These preliminary findings could guide clinicians and researchers in designing and testing musical activities for the therapeutic benefit in people with AD and their caregivers. And that's actually super relevant to me because I'm starting a postdoc that will use music and dance in people with AD and Parkinson's disease. So I should probably look up this paper. So that's it for papers on behavioral interventions, and last up we have 6 papers on physiological or medical interventions. As you'll see, this covers quite a broad range of basically any medical interventions other than pharmacological ones, which are covered in a separate episode. And I'll start this section off with one paper on using mesenchymal stem cells to see whether it can promote neuroprotection and functional recovery in AD. So this paper is called Feasibility and Efficacy of Intra-Arterial Administration of Embryonic Stem Cell-Derived Mesenchymal Stem Cells in Animal Models of Alzheimer's Disease. Oof, that was a long one. Okay, this was published in the Journal of Alzheimer's Disease by first author Kim and last author Lee. The authors investigated the neuroprotective effect of embryonic stem cell-derived, or ES-derived MSCs, so these are the mesenchymal stem cells, in cellular and animal models of AD. And they also looked at the safety of the intra-arterial administration of these stem cells in rats. They report that ES-MSCs displayed higher cell viability compared to those from bone marrow in amyloid beta-induced cellular models, and ES-MSCs were more effective in reducing intracellular A-beta levels. ES-MSCs also significantly inhibited A-beta-induced hippocampal cell death in a rat model of AD and promoted autophagolysosomal clearance of A-beta, thereby resulting in decreased A-beta levels in the hippocampus. The treatment additionally improved memory performance compared to rats injected solely with A-beta, and the intra-arterial delivery of an appropriate density of ES-MSCs was deemed safe and free from in situ occlusion or cerebral ischemia. These results suggest the therapeutic potential and safety of intra-arterial administration of these embryonic stem cell-derived mesenchymal stem cells for the clinical applications in AD. So the next 2 papers are on therapeutic delivery strategies using exosomes. So these are membrane-bound extracellular vesicles that have potential to act as a biological vehicle for drug delivery or genetic materials, particularly for crossing the blood-brain barrier and binding to the right targets. The first one tests a microRNA-based therapeutic approach in a rat model of AD. It was published in Frontiers in Neuroscience by first author Jahangard and last author Mola, and the title of this paper is Therapeutic Effects of Transplanted Exosomes Containing MIR29B to a Rat Model of Alzheimer's Disease. So the expression of the microRNA 29 family is significantly reduced in AD, possibly contributing to pathogenesis. This has prompted an investigation into microRNA-based therapeutic approaches, with an emphasis on the efficiency of microRNA transfer to the target cells. To this end, the authors transfected rat bone marrow mesenchymal stem cells and HEC293 T cells with recombinant expression vectors carrying either microRNA 29A or 29B precursor sequences. This successfully caused overexpression of AMiR29 and downregulation of their target genes, which include BASE1 and BIM, in the transfected cells. After confirming that microRNA-29, I'm referring to this as AMiR29 or microRNA-29, So after confirming that AMiR29 was packaged into exosomes and secreted from the transfected cells, the authors investigated a possible therapeutic effect of these engineered exosomes in reducing the pathological effects in amyloid beta-treated rats. These rats showed some deficits in spatial learning and memory, which were prevented by the injection of AMiR29 containing exosomes into the CA1 region of the hippocampus. These findings confirm the ability to package microRNA-29 in exosomes and suggest that these engineered exosomes show therapeutic potential for AD. Alright, so this next paper is also using exosomes, but this time it's for the delivery of P-glycoprotein, which is a member of the ABC transporter family situated in the blood-brain barrier, and it could play a role in amyloid beta clearing. So this was published in Neurochemical Research. The first author is Pan, the last author is Zhao. The title of this paper is Brain Microvascular Endothelial Cell-derived exosomes potently ameliorate cognitive dysfunction by enhancing the clearance of amyloid beta through upregulation of PGP in mouse model of AD. To be clear, PGP is the P-glycoprotein. So PGP expression is reduced in pathological blood-brain barrier, which could contribute to the impeded clearance of amyloid beta, or A-beta. Alright, pay attention to this next sentence because I'm not repeating it. The authors used human brain microvascular endothelial cell-derived exosomes inheriting PGP in the AD mouse model to test whether it could remove amyloid beta peptides from the brain by specific capture between PGP and amyloid beta. They found that these indeed facilitated the cerebral clearance of A-beta by effectively transporting A-beta out of the brain and improved the cognitive dysfunction in these mice. Okay, that was short and sweet, but a series of long, complicated words. So let's move on to a paper that investigates the literal fountain of youth, that is to say young blood. So they test whether using blood from young animals can improve the cognitive function of old ones by reactivating neurogenesis and restoring synaptic plasticity. So that sounds a bit morbid, but interesting. And the title of the paper is Young Blood Plasma Reduces Alzheimer's Disease-like Brain Pathologies and Ameliorates Cognitive Impairment in 3X Transgenic AD Mice. The first author is Zhao, the last author is Gong, and this was published in Alzheimer's Research and Therapy. So this lab tested whether intravenously injecting plasma from 2 to 3 month old wild-type mice into 16 to 17 month old 3X transgenic AD mice could have therapeutic effects on AD-like pathology and on cognitive impairment. This was done twice a week over 8 weeks, and multiple behavioral tasks were conducted after 4 weeks of plasma injections. The authors also assessed the levels of synaptic proteins and neuroinflammation using Western blots and immunohistochemical staining. They found that young plasma treatment improved short-term memory in the novel object recognition task and enhanced spatial memory and learning in both the Morris-Water maze test and the reversal Morris-Water maze test. In addition, the treatment reduced both tau and amyloid beta pathologies, as well as neuroinflammation in the mouse brain. That said, the authors did not find any significant changes in synaptic protein levels or in dentate gyrus neurogenesis. Overall, these results indicate that young blood plasma can improve cognitive function and reduce AD pathology in a mouse model, and it does suggest that blood transfusions could be a potentially effective treatment for Alzheimer's. But it doesn't suggest that it can reverse all signs of aging, so don't get any weird ideas here. Okay. This next paper is called High Frequency Transcranial Magnetic Stimulation Protects APP-PS1 Mice Against Alzheimer's Disease Progress by Reducing ApoE and Enhancing Autophagy. This was published in Brain and Behavior by first author Chen and last author Wang. So repetitive transcranial magnetic stimulation, which I'll refer to as RTMS, has been used in the treatment of AD, but the underlying mechanism is still unclear. The authors investigated the effects of a 2-week 5 Hz high-frequency RTMS treatment in 3-month-old APP-PS1 AD mice. They found that the RTMS treatment improved cognitive impairments and AD-like pathology, so this includes a decrease in phospho-tau, APP, A-beta, and PP2A expression. So those were reduced in these mice, and they also found that BDNF-TRKB signaling was significantly enhanced. They also found that the LC3-2 to LC3-1 ratio was elevated with a significant reduction of ApoE and of phospho-62 in the treated APP/PS1 mice. These results indicate that the therapeutic effect of RTMS may be in part through reducing ApoE expression and in promoting autophagic flux. So we're going to switch gears now to humans. This last paper looks at whether improving tissue oxygen supply could be neuroprotective, given that hypoxia might contribute to the pathogenesis of AD. This paper is called Hyperbaric Oxygen Ameliorates Cognitive Impairment in Patients with Alzheimer's Disease and Amnestic Mild Cognitive Impairment, or amnestic, sorry. Anyway, the first author is Chen, the last author is Li, and it was published in Alzheimer's and Dementia. So hyperbaric oxygen treatment improves tissue oxygen supply and ameliorates hypoxic conditions in the brain, and this study investigates whether it could be an alternative therapeutic for AD and amnestic mild cognitive impairment. I'm going to refer to this as AMCI. The authors recruited 42 AD, 11 AMCI, and 30 control AD patients for this study, and the AD and AMCI patients were treated with 40 minutes of hyperbaric oxygen once a day for 20 days. The control AD patients who did not receive hyperbaric oxygen treatment had similar clinical profiles as the treated AD group. Numerous neuropsychiatric assessments were taken before and at 1, 3, and 6-month follow-up after treatment, and a small group of treated patients were also assessed by PET imaging. In AD patients, 1 course of hyperbaric oxygen treatment significantly improved the cognitive function assessed by Mini-Mental State Examination, this is MMSE, and the MoCA test after 1 month follow-up, and the test scores were significantly better than in untreated control AD patients. The Activities of Daily Living scale was also significantly improved in treated AD patients after 1 and 3 month follow-up, and AMCI patients also showed increased cognitive test scores at 1 and 3 month follow-up. Lastly, in some of the AD and AMCI patients, hyperbaric oxygen treatment improved the reduced brain glucose metabolism as measured by PET imaging. Based on these and previous findings, the authors proposed that hyperbaric oxygen treatment may be a promising alternative therapy for AD and AMCI. So that wraps up the June 2020 papers on non-pharmacological treatments for AD. Be sure to check back here for upcoming episodes on clinical treatment of AD, as well as diagnostic tools and biomarkers, and we've also got a few episodes still on disease mechanisms. And as a reminder, if you would like to receive our bibliography, you can do so by sending us an email or by filling out the form in our episode notes. Alright, that's it for me, and speak to you again next time. Bye.
That's it for this episode. A huge thank you to the team that is working on sorting, summarizing, and scripting these abstracts, as well as the operations behind AMiNDR. The music is from Journey of a Neurotransmitter by Anusha Kamesh, musician and fellow scientist, and now member of the AMiNDR team. You can find the original piece and her other music on SoundCloud under Anusha Kamesh or on her YouTube channel, AK Music. Interested in joining the team? Give us a shout! We can always use help with content development, podcast editing, advertising, and you can be part of a new and exciting venture. Reach us by email at aminderpodcast@gmail.com or follow us on Twitter. Oh, we're also on Facebook now. Don't forget to subscribe to our mailing list if you want access to the bibliography for each of our episodes. The references come with timestamps. Timestamps.
so you can more easily locate the paper that caught your interest. Check our notes below for details on how to sign up. And very close to this, you'll also find a link to our feedback survey. Because, yeah, your feedback matters to us, so please, pretty please, let us know how we can make this podcast a better tool for you. And last but not least, thank you for tuning in with us. And on this note, we hope you found our podcast useful and accessible. Until next time.